Hi, I'm Hans Paerl from the University of North Carolina at Chapel Hill Institute of Marine Sciences, and we're here at Lake Taihu, China, studying blue-green algal blooms, and you can see the green color out here. It's very green and rich, and we've been working here for about 10 years doing nutrient limitation studies and also working on looking at sediment-water column interactions and the issue of internal versus external loading in the lake.
In numerous water bodies around the world, ranging from small lakes and ponds to large lakes, such as Lake Taihu in China and Lake Erie in North America, harmful blooms caused by cyanobacteria or blue-green algae are a major problem for the ecosystem services of drinking water supply, fisheries, and recreation. The harmful impacts of these blooms fall into four main categories: toxins, tastes and odors, effects on the aquatic food web, and biomass overproduction. The toxins are of special concern for water quality and are produced by many species of bloom-forming cyanobacteria. These include hepatotoxins that affect the liver, fast-acting neurotoxins which can cause death of livestock and pets that drink the untreated water, and toxins that cause skin reactions. Taste and odor compounds from cyanobacteria are especially a problem for drinking water supplies, but also aquaculture, and include the earthy or muddy tastes caused by geosmin and methyl isoborneol. Bloom-forming cyanobacteria also create problems for aquatic food webs, in that the colonies are difficult to ingest by filter-feeding zooplankton. They are a poor nutritional source for animals, and they outcompete phytoplankton species that provide higher quality food. And finally, there's a host of problems simply caused by the large amount of algal biomass produced during the harmful bloom. These range from clogging of water treatment filters to organic enrichment of the sediments, leading to anoxia and associated biogeochemical effects. This set of harmful impacts of bloom-forming cyanobacteria is only one of many reasons that phytoplankton ecology is such a major theme in limnological research. Phytoplankton are defined as the assemblage of microscopic eukaryotic algae and cyanobacteria that live suspended in the water column. And they are a major component of aquatic biodiversity. Phytoplankton are at the base of planktonic food webs and play major roles in primary production and biogeochemical cycling in lakes, ponds, reservoirs, and large rivers. Understanding what controls the biomass and community composition of the phytoplankton is therefore critical for our general understanding of aquatic ecosystems and our ability to predict what these ecosystems will look like in the future. Dr. Elena Litchman is Professor of Aquatic Biology at Michigan State University, and here introduces the trait-based approach towards understanding phytoplankton ecology.
The trait-based approach provides a mechanistic framework to help us understand phytoplankton community structure and its responses to changing environmental conditions. By traits, we mean any heritable feature that can be measured at the individual or population level. These features may be morphological, for example, cell size, physiological, for example, growth versus temperature characteristics, or life cycle features, such as resting cells, sexual versus asexual reproduction, and others. There are often large differences in traits both among and within phytoplankton taxonomic groups. These differences can affect how growth and loss rates change in response to seasonal or longer-term variations in the environment, resulting in large changes in phytoplankton community structure over time and space. Many traits of species also exhibit large intraspecific variation, and this variation, along with new mutations, provides the basis for ongoing evolution and adaptation. Building on work by Ramon Margalef, Colin Reynolds, and others, we have been developing a trait-based typology where phytoplankton traits are classified by their ecological function, such as resource acquisition or reproduction, and trait type, such as morphological, for example, cell or colony morphology, physiology, and so on. Many of these traits can vary within a single species according to environmental conditions, and even among individual cells. For example, our recent work led by Arno Luchar and Dedmer van der Waal from the Netherlands on a strain of bloom-forming cyanobacteria isolated from Lake Taihu in China showed that traits varied according to light availability, nutrient availability, and CO2 conditions.
The population size of each species of phytoplankton in a sample of water is the net result of gain processes, notably growth by cell division, but also transport and migration, minus population loss processes, such as grazing, parasitism, and sinking. There are large trait differences among algal species and among algal groups in their growth relationships with environmental variables, and in their susceptibility to loss processes. Cellular growth rates are usually expressed as the exponential rate of increase in cell concentrations, specific growth rate with units of days to the -1, and are primarily a function of nutrients, light, and temperature. There are large trait differences among algal species and groups in their growth relationships with these environmental variables. Nutrient uptake and growth rates are often a saturating function of nutrient concentrations, and the parameters from these curves can be considered phytoplankton nutrient traits, which differ greatly among species. For example, small cells such as cyanobacteria have high affinities for nutrients, low Ks values, and are efficient in low-nutrient oligotrophic waters. Many species, notably bloom-forming cyanobacteria, are capable of taking up and storing nutrients well in excess of their immediate needs. This luxury uptake of nutrients may then allow cells to continue growing on their stored reserves, even when they are transported into lower nutrient waters. These transport and migration processes also mean that algal blooms may accumulate in areas that are well away from the location or depths of their source populations and nutrients. Another set of traits that contributes to the success of certain phytoplankton species, called the mixoplankton, is their ability to use dissolved and/or particulate organic materials. These materials are a source of nitrogen, phosphorus, and other nutrients for algal growth, as well as provide a supplement to the carbon and energy acquired by photosynthesis. The light traits of phytoplankton include those apparent from growth versus irradiance curves. These typically show a rise towards saturation, and for many species, depending on their physiological state, inhibition of growth at highest irradiances. Many cyanobacteria have a high alpha, the initial slope of the growth versus irradiance curve, meaning that they grow efficiently at low light, while many green algae grow well in bright light conditions. Phytoplankton also vary greatly in their growth temperature parameters. With cyanobacteria, even isolates from cold polar waters generally showing higher temperature optima and thresholds. Phytoplankton also show large differences in traits that affect their susceptibility to loss processes such as grazing and sinking. Grazing protection may include cell size, colony formation, spines, and the production of antagonistic or allelopathic chemicals. While traits to reduce sinking losses include motility and features that are parameterized in Stokes' law for sinking velocities in a viscous medium. A fundamental question that has long fascinated ecologists is How can it be that so many species of phytoplankton co-occur in the same volume of lake water? Ecological theories predict that only a limited number of species can stably coexist at equilibrium, given the competition for limiting resources, yet lake water samples often contain dozens of species. In his classic paper, G. Evelyn Hutchinson referred to this as the paradox of the plankton. Subsequently, theoretical and experimental studies have shown that many species can coexist on a few limiting nutrients when resources fluctuate, and when species have non-linear responses to resources. Spatial variations in resource availability and loss processes may also play a role in supporting this diversity. Cyanobacterial blooms can cause drastic decreases in phytoplankton diversity that in turn reduces the quality and availability of food for the zooplankton. Variations in environmental variables such as light, mixing regime, temperature, and nutrients, and in loss factors such as grazing, cause changes in phytoplankton biomass throughout the year. The interactions between species traits and these seasonal forces give rise to phytoplankton succession. That is, the changes in phytoplankton species composition throughout the year. In cold temperate lakes, diatoms often become dominant during spring and fall mixing, while cyanobacterial dominance is often more apparent in summer and early fall. However, patterns vary widely, especially among lakes of different latitudes and different climate regimes. The high temperature optima for many, if not most, cyanobacteria has raised concerns that global warming may promote the growth and dominance of noxious bloom-forming species. Additionally, climate change may cause greater stratification that favors buoyant, bloom-forming cyanobacteria and may lead to prolonged anoxia and increased internal nutrient loading. More frequent and extreme storm events in a warmer climate will result in increased runoff and increased external nutrient loading, which could also promote cyanobacterial growth. In addition to their occurrence in warm lakes, bloom concentrations of cyanobacteria have also been recorded in certain cold water environments, including cold, deep Great Slave Lake in subarctic Canada, but mostly in warmer inshore locations. These observations raise the question, how important is lake water temperature relative to nutrients for cyanobacterial bloom development? Dr. Sylvia Bonilla is a professor in the Limnology section of the Faculty of Sciences at the Universidad de la República in Montevideo, Uruguay, and here describes a collaborative project throughout the Americas to address the question of environmental controls on cyanobacterial biomass.
In this study, we assembled a large database of phytoplankton species abundance and associated limnological data from published and unpublished records. The data encompassed observations from 464 lakes and cover an extensive range of water body sizes, depth, and elevations, as well as trophic conditions. There were no clear trends in cyanobacteria biomass as biovolume concentrations along latitude or climate gradients, with the exception of lower biomass in oligotrophic polar lakes. Phosphorus was the primary resource explaining the variations in cyanobacterial biomass, while nitrogen was also significant, especially in shallow lakes under 3 meters depth. Despite the large climatic gradient, cyanobacterial biomass was only weakly correlated with water temperature. Our study shows that unwanted cyanobacteria can develop high biomass in any climate if nutrient concentrations are sufficiently high. Solutions toward managing harmful cyanobacteria should thus emphasize nutrient control irrespective of temperature regime.